Okay, a warm welcome to all ISPIC participants and teams again. This is the fourth webinar of the ISPIC 2021 Inspirational Webinar Series focused on the untapped potential of fungi and alternative proteins. So before we engage with our speaker and move into the presentation, a few rules of engagement. So you as a participant can ask questions. Please use the Q&A button for the questions to the speaker, but also feel free to use the chat. And again, iterating this once again, please ask any questions which come to you guys. Also, some of the questions which were asked in the other webinars, but were not relevant to fermentation. This is the opportunity to ask those questions again. And after the webinar, we'll have a specific dedicated Q&A session for 30 minutes so that participants can still ask their questions using the Q&A button, or they can unmute themselves. Please raise your hands using raise your hand button and we'll unmute you to ask a question live. So moving on, today we have Rama Tentu with us as a speaker on this topic. And Rama Tentu has a master's in chemical technology from IIT Delhi, and previously she has worked with GFI India for over a year as a research associate. During her time at GFI, Rama has been instrumental in building ties with several key institutions in IITs across the country, and has led several key projects such as the fungi and microalgae and seaweed strategic analysis, and the texturization technologies strategic analysis as well, which I'm sure most of you or most of the participants in this webinar have familiarized themselves. So currently Rama has moved to the US, and she's pursuing her MBA at Yale University. So a warm welcome to you, Rama.
Nice to meet everyone.
Thank you. So next is just focusing on some of the generous support which we are getting from our title sponsors, key sponsors, organizing partners, supporting partners across both corporate as well as institutions, universities across India. So this would not have been possible without the support from all these sponsors as well as partners. So moving on into what were some of the problem statements, I think, which we generally discussed in our final webinar, where we deep dive into some of the RFPs. So we have, specifically in terms of fermentation, in track 1, 5 RFPs focusing on various areas, including fungi. So for today's webinar, I think people who are interested in RFP 1, that is basically screening of filamentous fungi to identify new candidate strains or strain improvements for enhanced protein and organoleptic properties, I think the candidates or participants can gain insights and ask questions about. The second RFP as well, because it focuses on precision fermentation, that is, let's say, microorganism non-specific. And I think you can use fungi as well to do precision fermentation and derive proteins of interest, which is applicable to the smart protein sector. The third RFP is basically tapping into biomass fermentation. Again, fungi is something which is commonly used in terms of biomass fermentation. So I'm not going to the details of these RFPs, but these 3 RFPs would be the ones which candidates will have an opportunity to ask relevant questions about in today's webinar. The other 2, we have scheduled a couple of more webinars. Please stay tuned in next week for microalgae and seaweed, and also in terms of basically feedstock optimization. So track 2 participants, just reiterating this again, you guys are free to formulate your idea in terms of starting your own company across the value chain area of fermentation, feedstock optimization, strain development, fermentation, bioprocess design, and even final product formulation. So obviously you have a ton of flexibility in terms of your ideas and where you want to actually plug in. So like I said before, Q&A session will begin, at the end of the session or the webinar from Rama, we have 20-30 minutes of Q&A. So please feel free to submit your questions again, and please do not hesitate. Now the floor will be, I open the floor for Rama to basically present her webinar. So over to you, Rama.
Thank you, Akshay. Glad to be here. Let me quickly share my screen and let me know if you're able to see.
Yes, all good.
Great. So thank you, Akshay, for the generous introduction. I'm glad to be here. Last year, I was there when the challenge was happening. So really excited to see how the different tracks have shaped up and how we have so many participants interested in very specific scientific challenges. As Akshay mentioned, the focus of today's webinar is on applications of fungi. And I just want to mention that last year when we were doing this analysis and trying to study this space, it was still an evolving sector and did not have like, I mean, there were companies out there, but there wasn't as much hype as there is now. People have started recognizing how important this microorganism is for the alternative protein sector. So we'll be seeing some of the examples, but I'm sure like there are many more companies which have now come up, which only demonstrate that what we had predicted this as being a very important platform for alternative protein sector. So with this, I'll just give an overview of the different topics we are covering today. So I'll give a brief landscape of alternative protein sector. I'm sure you have already heard this 1,000 times, but just to build context in terms of where does fungi fit in this sector, what does fungi mean? What are the current use cases of fungal fermentation? How does the value chain look like in terms of starting from a strain to how do you create a product? And finally, looking at the current application areas. So there is not just one application, there are many applications, and you can bucket them into different categories. And lastly, opportunities across the value chain. So what are some of the untapped opportunities for uh, for 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 participants like you to focus on? What are the different areas you could explore or are already exploring? With this, uh, you already know, you have already seen this chart, but again, reiterating some of the important points here. You have three different categories of plant-based alternative protein technologies: plant-based proteins, fermentation, cultivated meat. They basically differ on how you are producing the protein that is used for creating these meat analogs. And today we are going to focus on fermentation, but in case of fungi, it can actually cut across applications in plant-based proteins, cultivated meat. So although you use fermentation as a platform for producing fungi-based compounds, you can use these in plant-based meat or
I'm not sure because I got disconnected.
yeah, it seems I too got disconnected.
Yeah, if you could please share your screen again. I think we have lost your
Yeah.
screen as well.
Yeah. Also, I think we should. Yeah. Can any participant confirm they are able to see my screen?
I'm able to see it, Rama, so probably everyone else should be able to see it. Shadul?
Okay.
Yes, I can see it as well.
Okay.
Receive confirmation from
Okay.
the participants.
So I'm not sure where I got disconnected, but I was talking about how the applications of fungi are not something which is newly discovered. It has been used historically in various food products, and some of the examples include cheese, beer, wine, making bread, kimchi, tempeh, producing enzymes and food processing aids, vitamins, natural flavors, and pigments. And in the 1985, even like filamentous fungi was used to create meat analogs, although it not pick up, but it was already there in the market. So this is just to demonstrate that there are fears that people have around, oh, is it safe? Isn't it a mold? How can we eat this? So in that sense, fungal fermentation is nothing new to the food industry, and there already exist strategies in which you can make these foods safe. So it's about figuring out how you can make novel products at this point of time. And in terms of alternative protein sources, if we look at the different types of sources we have, so a lot of us have seen products which use legumes and pulses as sources of protein. So be it soy protein, pea protein, chickpea protein. Uh, Now people are also looking at rice, wheat, and maize protein for better functionality. And then there are like less used protein sources like quinoa, amarnath, which are pseudo cereals. Nuts and seeds are also like other sources of protein. So these are mostly plant-based proteins which have been traditionally explored. But now there are emerging sources of proteins like microalgae, seaweed, fungi, uh, and also cultivated wheat, which is like animal cell culture. So the idea is that as we diversify the sources that we look at to extract different kinds of protein, it gives us different functionalities and different kind of properties. So it's important to look at these sources from a nutritional perspective and also from the perspective of providing various types of taste profiles. So before we dive into it, like this is supposed to be a poll question, but like you can just drop your answers in the chat. So is it fungi? Like, how do you pronounce this word? Like, is it fungi or fungi or fungi or fungi? Anyone who knows about this? Okay, I understand it's a Saturday evening, people are tired, and some people are leaning towards fungi. It's fungi, and I apologize in advance for pronouncing otherwise. It's hard to keep track, but this is so. Just remember ghee, and like it's usually pronounced as fungi. Um, Now, what really is this microorganism, and how do we understand what are the thoughts that come to your mind when you think about fungi? So this is a classic example from our childhood stories where there are 5 or 6 blind men trying to figure out what an elephant is, and they touch different parts of the elephant, uh, and that's how they try to say like what this elephant is. And the same is the case with fungi, because this microorganism, as I said, has so many applications, and depending on how you look at it, you could like the first thought I think that a lot of people think about when they think of fungi is it's a pathogen. It's something. It is a disease causing organism. It spoils your food, and it can poison you and whatnot. So yeah, definitely one of its existence in this nature is in form of a pathogen. It is also a medicine, and if you know, penicillin and a number of antibiotics are made out of uh, extracellular enzymes secreted by fungi in bioreactors. So it is a medicine as well. It's also a killing machine. You can see that there's a dead ant over there, and it's actually been taken over by a certain species of fungi. And yeah, so it could be a killing machine if you're an ant in your next birth. And it does make food better. We all like know how yeast is used for fermentation of grapes, and how it is used to make wine, beer, spirits, and also bread, for example. And today, as we are discussing, it's an alternative protein. So really this microorganism, I feel, is a work of art, can do so many things depending on how you want to use it. And looking more scientifically into what actually this microorganism looks like at a microscopic level, before we look at the microscopic level, I think all of us have at some point in our high school studied that fungi is a heterotrophic organism, doesn't need sunlight to grow, but feeds on carbon and organic matter uh, to grow itself. It's a eukaryotic organism, meaning that it has a nuclear membrane, cell membrane, and nucleus, a well-defined nucleus. It the second thing which probably like people get wrong is like some people would say that it's unicellular, other would say it's multicellular, but really it can be anything. It could be unicellular and multicellular, and the same organism can sometimes become unicellular and multicellular, which is very interesting. And if you look at uh, if you look at the diagram here, there's a unicellular fungi, and something like a yeast is a very good example of unicellular fungi. And then there is filamentous fungi, which is microscopic, and this is typically the kind of fungus you see on bread in the form of molds. And I'm sure you have seen these strands and this amorphous structure. And then there is lastly mushroom, And this is what we see as like the fruiting bodies of mushroom, and mushrooms are nothing but a collection of filamentous fungi. So again, like these are multicellular organisms, both of these, and this is a unicellular organism. And in terms of a life cycle, for example, this is for life cycle of mushroom, and a mushroom like really generates from like spores, and then these spores grow into hyphae, and hyphae grow into mycelium, and then like they form well-defined structure which look as fruiting bodies, but underneath it's just like well-connected tissue, well-connected hyphaes and mycelium. So just to clarify, like all these strands that you see which are really well connected are known as mycelia, and a lot of times in alternative protein sector, you would hear the word mycelium-based fermentation when they're actually referring to filamentous fungi as sources of protein. An important property of fungus is that they first digest the food and then ingest. What this means is that uh, like humans would eat the food, and then like the enzymes in our stomach and our intestines will break down this food. But in case of fungus, they would release enzymes outside their body, and that would break down the food outside their body. And then once the food is decomposed, they eat that food, like they consume that as food. So that's an important property, because that's how pharmaceutical companies have been able to uh, extract extracellular enzymes and use them for production of various pharmaceutical compounds. And yes, just as I mentioned, so there are three big buckets of fungus that you could look at: yeast, which is unicellular; mushrooms with fruiting bodies; and filamentous fungi, which is like more microscopic in nature. And I urge you to think of some examples of fungi in our daily life, and probably I will not spend time on this because I've already talked about this in the last slides. But basically, like a lot of our antibiotics come from fungus, which is filamentous fungus grown in bioreactors, and then their extracellular components are extracted out of it. Um, I mentioned about corn. Corn has been using filamentous fungi to make mincemeat products, as well as um, meatless nuggets, for example, and they have been in the market since the 1990s. They're used for making enzymes. These enzymes can be used in the food industry, it could be used in the cosmetics industry, and as I said, it also has medical applications. Less known applications would be, so these tables, like these tables and chairs, whatever they are made out of um, mushrooms actually, and people have been able to figure out how to grow mushrooms in a way that they can take a particular form and shape. We have a number of companies who are working to make animal replacements of leather. So mycelium-based textiles have really picked up, mycelium-based shoes. So a lot of companies are now working on not only like food replacements, but also like replacements of products used in our everyday life, including things like building materials. So there has been a building which is made out of mushroom. If anyone is interested, they can look up that online. Coming back to the food sector and the alternative protein sector, as you're already familiar with what fermentation means. So fermentation is basically cultivation of any microbial species, either whole biomass, for whole biomass, or to extract certain components out of it. And while, as I said, fungi itself has been well established, fermentation has also been a well established technology, but the application in the fungal protein sector, the alternative protein sector, is relatively new. And I think this is a graph that you might have already seen, but again, important to understand that there are three ways in which you could leverage fermentation as a platform. So you could use fermentation to do traditional fermentation, which is basically use these microorganisms uh, in a whole biomass format, and use them to transform ingredients and improve the quality of ingredients. you necessarily don't consume, like use that microorganism. You're not consuming that directly, but using it to improve the quality of other ingredients. Then there is biomass fermentation, which is basically for the purpose of consuming the microorganism itself. Obviously, the microorganisms is deactivated and made safe to use, but the idea is to use it as a protein source or a source of protein isolate, lipids, fats, etc. Then there is precision fermentation, which is basically uh, you can do precision fermentation for traditional fermentation and biomass fermentation as well. But the idea here is that you are tweaking the DNA and genetic makeup of the microorganisms and exploiting that to create desirable traits, which could lead to like formation of specific molecules and compounds, or also like to achieve certain growth rates or productivity. Um, and just giving some examples of how these have been leveraged so far. So traditional fermentation has been so there are companies like so you can see the name of these companies and look up Planetarium's Mycotechnology, Miocos. So the idea here is that, for example, mycotechnology uses uh, shiitake mushroom filamentous fungi to ferment rice and pea protein. So they are improving the quality of rice and pea protein using this mycelia. So as I said, the mycelia excretes enzymes extracellularly, and these enzymes then break down the protein components or break down the unflavored, undesirable flavor components within the rice and pea protein, making it more functional, making its organoleptic properties better. So all these companies have been working in this space and specifically looking at leveraging fungi as a platform. Then there are companies which are utilizing fungi to create whole biomass products. So uh, you have almost all of you must have like seen that most of the products out there are chicken nuggets or mincemeat products, which are basically they don't have a long range structure, and they don't have like in the market, if you were to go and buy uh, you know from a butcher chicken, and it would have a certain like structure to it, and that is something we have not been it is not common to see in terms of the products that we buy. And all these companies are trying to mimic that structure of meat. And as you can see in like this photograph by Meaty Foods, and they're again using filamentous fungi to create this long range structure and like the juiciness profile, the textural profile of meat. Quan is attempting to do this, and then Nature's Find has used a certain type of um, fungal species which they extracted from the Yellowstone National Park, so extremophiles, to make this. And then we have Atlas Food also doing a similar thing, and Mycorrhiza, which has had last year released these Swedish vegan balls. And I could talk a little more about it because they have worked on feedstock optimization very well. They recently also released fungi-based fats. So that's a very interesting application. It's the world's first application of fungi to create um, fats, particularly for the alternative protein sector. Then there's precision fermentation, which is basically changing the genetic makeup of the organism to get the functional and sensorial profiles necessary for use in alternative protein products. And Clara Foods has done strain optimization, identified species which can give you organisms which can produce egg white proteins. Perfect Day again doing this for milk proteins, for casein and whey. You already know probably about Richco Life Sciences, which is now Lazarus Bio, how they're using microorganisms to produce growth factors, which then go into cultivated meat applications. So I'm sure you have already seen that uh, how there are effects of fungi, which like impact of fungi on both like the fermentation, the plant-based space as well as the cultivated space. So this is going to be an important driver for improvement in the quality of ingredients that we see out there. In terms of recent activity in this space, I mean, there are many companies who have shown interest, and like there has been good funding that has been coming into the sector. For example, Unilever recently announced partnership with Enough, which used to be called as 3F Bio, and they're basically partnering to scale up. Enough recently opened their manufacturing facility um, in the Netherlands, and Mycorrhiza obtained like $9 billion of pre-seed funding. I think they raised another round of funding between the time I put this slide and now. Nature's Find received no questions asked letter from FDA in response to their grass safety determination, showing that like there has been more regulatory acceptance. Fuming Ingredients raised €500,000 of investment from AB InBev, and they're also scaling up their operations using AB InBev. So yes, a lot of activity in this space. And now let's talk about what does it take to make like these products. What does it look like scientifically to take a strain and then make a alternative protein product out of it. So in terms of production process, there are three main steps, and this is kind of common with any kind of fermentation process. And I think you will also look at this same graph when you look at microalgae and microalgae particularly, and any microbial protein source. So it could be bacteria, it could be microalgae, it could be fungi. And when we look at each of these categories, with like first step is always strain selection. It is about identifying the best strain which you will go ahead and scale up eventually. And this is obviously a critical step because it determines the properties of the derived ingredients. It impacts the downstream processing in terms of what are the side streams produced, what is the growth rate, what is the productivity, etc. So strain selection is a very important component, and I'll talk about it in the coming slides as well. Then I see Devika has raised hand. Is there? Okay. I will continue. And the second step, once you have identified the strains to use, is like obviously like producing it in a bioreactor. And at this stage, you you should obviously know whether you are doing liquid state fermentation, semi-solid state fermentation, or solid state fermentation, and what type of organism you're using, yeast or filamentous fungi. And depending on what type of fermentation you're doing, your bioreactors will look different. Your kind of feedstock that you're using will look different. And whatever feedstock you're using, whatever kind of reactor system you're using, the important step here is to optimize the nutrients that you're giving to this organism, so that there's a certain productivity and growth rate that you achieve. Also, bioreactor design is equally critical in terms of determining how efficient it is and how easy it is to grow the microorganism and eventually extract components out of it. And once you've like grown like the microorganism in a bioreactor system, you then move to like downstream processing, which is about how do you harvest this. So harvesting basically means that once you grow filamentous fungi in a bioreactor, it would probably have like 1% concentration. So for every 1 unit of biomass which is present in the reactor, there will be 100 units of liquid present in the reactor. So how do you separate this? How do you concentrate the solid components? And then also like looking at how do you remove RNA and sterilize this biomass, because whenever you have these high growth microorganisms, they have a high RNA content, typically in the order of 10%. And human body cannot take I think it's 2% RNA, which is the limit to how much RNA we can digest. And because of that, like there could be toxicity issues if you consume this. And hence destroying the RNA is an important part of downstream operations. And again, you need to look at what organism it is. For mushrooms, this may not be applicable, but this is applicable for yeast and filamentous fungi. And finally, once you've like made sure the safety aspects of it, you have concentrated your microorganism, looking at how do you extract functional ingredients from it. If you're making whole biomass, you probably don't need this, but if you're extracting proteins, enzymes, cell wall, cell wall components like chitin, you would need to think about the downstream extraction process, and I'll explain what that means in the coming slides. Again, trying to emphasize how the liquid state fermentation looks different from solid state fermentation. So for liquid state fermentation, the typical steps would include strain selection, selecting bioreactors, selecting growth media, doing RNA reduction, because in liquid state fermentation, you will be mostly using yeast and filamentous fungi to grow the microorganism. So you need to do RNA reduction, you need to do harvesting, concentrate the biomass. Again, like typically concentration happens in multiple steps. So one step is like separation, and the second step is concentration, and then you will have multiple ways to keep concentrating the biomass. Then you would need to do post-processing in terms of extracting specific components. And once you have extracted, let's say, protein isolates, you would need to like feed it into product development, and that in itself is a separate process. Solid state fermentation, on the other hand, is relatively simpler, especially for mushrooms. You already like have a set. There's a lot of literature on what different types of mushroom available in the world, and you could again select the type of mushroom you want to use for production, and the production will typically look like a normal production for mushrooms. So um, the way mushroom is produced for our everyday life, it will probably look like this, but then there's scope for innovation in terms of the kind of fruiting bodies you can make. So earlier I showed in a slide how leather made from mushroom looks like, right? So how do you create big slabs of mushroom? We have seen these small uh, small button mushrooms, right? You can get slabs of mushroom, and that completely depends on how you grow them, what kind of nutrients you give them, what types of strains you use, and what kind of conditions you use. So controlling humidity, temperature can all affect how a mushroom grows and what is the structure of the fruiting body. So once you've cultivated this mushrooms in a certain setting, you harvest them. Because it's solid state, it's easy. You just have to like take out the solid components and make sure it is clean and sterilized. And once you have the solid components, product development is all about like recipes and working with chefs to understand how you can incorporate them into making alternative protein products. Some of you might be thinking, how can a mushroom give a property of a chicken like meat? And the answer to that is strains. Most important thing is strains here, because there are a lot of mushroom species, and this is something I describe in my report as well, which have the taste of a chicken. So there are mushroom species which are called like chicken of the woods, or bacon of the woods, and things like that, and they really taste like mushrooms, and that's how people have been able to exploit it. And the second component of it is obviously solid state fermentation. Is how do you make long range structures out of mushroom, which give them the whole muscle texture, and make them good candidates for uh, creating these meat analogs. Now, having described that, I will quickly walk through what each of these steps which I described here look like, and what are the important components to focus on. So when looking at strain selection, the idea is to identify efficient strains which give you high growth rate, high protein content, have a history of safe use, have are able to utilize the growth media in an efficient manner, do not produce toxins, and allow for easy extraction processes and harvesting processes. So this is what we mean when we are saying that we need to screen for efficient strains. And efficient strains can be made either through genetic modification, as in case of precision fermentation, and there are a lot of techniques for that. And you can also not genetically modify in an artificial sense, but allow for natural mutations uh, through uh, through certain techniques, and that leads to genetic modification, but not in a classical sense. So some of the regulatory authorities may call it a non-GMO approach, but then the idea is to create a lot of genetic mutations, and then you screen from a number of genetic mutations to see which ones are producing the most efficient strains. Then the next step is, as I said, you have to figure out what the growth media that you're using is efficient for that microorganisms and vice versa. So what this means is that you're not using very costly feedstock, because a lot of companies which are now working on filamentous, producing proteins using filamentous fungi, is figuring out how you can use low cost feedstock, how you can use side streams, because this impacts the overall cost of the product. So it's very critical to bring down the cost of the substrate. Also, efficiency and productivity is naturally important, because if the nutrient uptake is not efficient and it doesn't translate into biomass productivity, it is not a useful feedstock to have. So again, this is an important component. uh, I have skipped a slide on bioreactor design, but depending on whether you do solid state fermentation or liquid state fermentation, the choice of bioreactor is also very important, because it impacts how easy it is to grow and how easy it is to maintain the structural components, structural nature of the microorganism itself. So, for example, if you use a bioreactor which has a very high uh, uh, stirring capacity, it might destroy the microorganism. So the microorganism might not just grow. So all those things are important. And at the same time, the reactor conditions are important, and that is where bioprocess design comes into picture. So once you like uh, optimize for biological parameters, which is identify the right strains and substrate, which is what we discussed in the previous slides, you need to look at within the bioreactor, what is the moisture content, what is the pH, what is the temperature, what is the gas phase, liquid phase composition, aeration, particle size, etc. All these are important physiochemical parameters which determine not only what kind of bioreactor you should choose, but also, like, once you have chosen, what is the exact range in which all these physical parameters need to fall into in order for getting optimal production. Last but not the least, like I mentioned, like mixing and agitation are important mechanical parameters which determine the biomass growth rate, but also like how like whether you are able to keep the microorganism alive in those specific conditions in which the bioreactor is operating. So once, like, again, like moving downstream, uh, which is basically how do you extract components out of these microorganisms. Again, it depends on what you're targeting. So you could target extracellular components or intracellular components. So as I mentioned before, like after you have produced the microorganism in the bioreactor, you need to harvest them, concentrate them. And then if you have to look at intracellular components, you have to do cell disruption. If you're looking at extracellular components, you need to look at the media in which the fungi has grown. So focusing first on cell disruption, which is intracellular components. Once you do cell disruption, the next step is obviously like separating the intracellular components, removing the cell debris, getting the intracellular product, and then concentrating it. If you're looking at spent media, which is the enzymes or extracellular component of it, you do selective separation, concentrate it, and then purify that particular extracellular product. So again, this is energy intensive, it's cost intensive process. And but for high value components, this can be a very important step and can actually have other applications other than what we have seen. So these applications can go into cultivated meat for creating growth factors. It can go into improving the quality of pea or rice protein, for example, as I showed for mycotechnology. So again, downstream operations can also happen in isolation, can be done by a different company, but again, it has a very different application as compared to like the upstream operations. I'll skip this question and just get to the different application areas in the alternate protein sector. And many of you have now been familiarized with all the examples I've been giving. And so what do these different applications look like? You have yeast and mushrooms. Yeast has been, for example, used for making wine and alcoholic beverages, and basically it acts as in the traditional biomass fermentation sense, but you can also extract protein out of it. Same is true for mushrooms. You can consume mushrooms directly, but then you can also create protein isolates from mushrooms. Then filamentous fungi, we have seen a lot of examples now, which is like creating meat analogs from filamentous fungi. And basically, because it's filamentous in nature, once you concentrate this whole biomass, they give the meat-like texture. And this is like the most commonly known application as of now. Then there is mushroom whole cuts, which is what I was talking about earlier, is how do you get from a fruiting body of a button mushroom to something like this, which is slabs of mushroom which resemble a whole cut meat. And this is a relatively new application, and Atlas Food is one of the pioneering companies. They have a wealth of knowledge on what kind of mushroom strains to use, how do you manage productivity, growth rates, reactor conditions, such that you can have these slabs of mushroom. Then there are flavor components you can derive, and yeast has traditionally been actually used to derive these flavor components. So yeast has an umami flavor, and if you have used any animal-based meat packed ready to eat products, for example, in Asian countries, especially like in Korea, China, etc., people buy chicken broth, meat broth, etc. And those broths are additionally flavored with yeast derived flavors because they have a very close flavor uh, to these meat products. And big companies like Angel Yeast have been playing in this sector for a very long time to make these flavors. Then mycelium fermented proteins, I have explained to explained this before, which is improving the quality of other plant-based proteins using mycelia. And this is again something that microtechnology has done, and I explained how pea and rice protein have been improved using shiitake mushroom filamentous fungi. Last but not the least, there is another mycelium application that we see is in production of scaffolding materials. So these could be used. So one is mycelium can be used to make scaffolds. So you can give them a particular direction to grow, and then they make these scaffolds called cultivated meat applications. And second is that you use the cell wall component of fungus, which is called chitin, and this chitin can be extracted and then used to create my scaffolding materials. So again, like these are applications that we see right now. Some of them have been underexploited. For example, yeast and mushroom protein isolates. Very few companies are working on this. Mycelium fermented proteins also, like few companies are there in this space. Mushroom whole cuts, few companies are there in this space. So a lot can be done here, and a lot has been happening in particularly filamentous fungi space. So yeah, this covers the entire application areas that fungi can touch upon. And again, putting this in text format mostly. So whole cut meat analogs, we have these companies like Atlas Food, Texture meat analog 3F Mycovation is based out of Singapore, India. Protein brewery, Quan, Mycorrhina, the companies working in this space. Protein isolates, not many, but yeah, there are some of them. Phototech, more foods, Fumi ingredients. Not most of them don't have products in the market right now. Flavor compounds, Angel, Yeast, Biospringle, Lasafre, all these companies, most of these companies have been in the space for a long time, but there are new companies like MedFood Lab, which recently came up in Israel. Scaffolding materials, Excel Scaffold, this is probably the only company in this space right now. Microtechnology, probably the only company in fermented protein space. So a lot of people have started working here, and I hope some of you find the inspiration to do the same. We have discussed this, and again, like reiterating the kind of opportunities we have is across the value chain: strain selection and optimization, feedstock optimization, identifying what kind of bioreactors to use, how do you optimize the bioprocess components, uh, how do you identify improvement in organoleptic properties using these fungi, and how can you use them as ingredients in plant protein applications or cultivated meat applications. Protein extraction techniques, I think, will become more relevant when people start looking at mushroom derived proteins as well as fungi derived proteins. And product development, obviously, uh, this is an ongoing process, important for any kind of um, any kind of ingredient you're looking at, be it protein isolate, whole cut, mushroom, or flavor compounds, etc. And particularly in India, we have the opportunity to leverage solid state fermentation for protein production. There are a lot of smallholder farmers doing mushroom cultivation. We have many research centers working on identifying mushroom strains, but for different applications. And so there is an opportunity to leverage this and venture into this and basically create a decentralized system where once you can create an IP around how do you cultivate this mushroom and like allow farmers to actually grow these. Um, then there is like this is a very obvious application, which is low cost fermentation, and India is the uh, leading force in like, for example, generic drug manufacturing, and that's precisely because we have the capacity to be able to bring down the production costs of uh, uh, traditional fermentation processes. And particularly for alternative protein sector, this means bringing down the cost of feedstock, and this means using indigenous feedstock. Can we use molasses, for example, which are byproducts from the sugar industry, and other food waste that is generated in agricultural processes? We have very strong biotechnological expertise, and then optimizing for bioprocess design, which can further bring down the cost, and also the infrastructure as well as biopharmaceutical expertise that exists in India. So yes, so there are opportunities in this sector, and then there are specific opportunities in India per se. And with this, I'll encourage you all to look at this sector. It is going to be the next plant-based for the alternative protein sector. And uh, with this, I will probably open for questions.
Yeah, thank you very much, Rama, for that wonderful session. We have a ton of questions in the Q&A section, so probably we can go one by one. So Arman is asking, can non-GM approaches be implemented to increase protein content in strains of filamentous fungi?
Yeah, I think I partly answered that during the webinar. So you could use naturally induced mutations like through UV radiation or chemicals. So you basically take a set of microorganisms, expose them to all these external uh, uh, external stimulators, which could be radiation or chemicals, and these lead to mutations which uh, which will be like a large number of mutations. What you would need to do is, like, let's say you have like 1,000 different types of strains which generate as a result of this, then you do a high throughput screening and identify the strains that are most relevant for you. So this is a process which has been like under certain circumstances been called non-GMO. Like, technically anything that leads to modification of DNA is a kind of genetic modification. We are all genetically mutated human beings. We have evolved over many years. But technically, what I'm explaining in terms of naturally induced mutations, or even artificially induced mutations without performing any of the genetic modification techniques, would also come under non-GMO techniques. Actually, am I missing the word for this? I feel like I am. There is, I think, a word for this.
For which one, Rama? For non-GM approaches?
Yes, I mean, the one that I just described, which is like chemically and mechanically induced mutations, and then doing high throughput screening.
Yeah, I mean, yeah, I think both of them are like related. For example, probably doing a high throughput, let's say, analysis and performing these mass mutations together could be probably a way to circumvent GM, for example. And okay, sorry, let me. Yeah, can mycelium be a nutritive source for meat logs instead of the fruiting body? I think the answer is yes, but if you want to answer this question, Rama.
Yeah, I mean, technically anything could be a nutritive source for plant protein. I mean, any organic matter. But particularly for mushrooms, like, you would need to do some kind of extraction processes. I think direct utilization will not probably benefit as much as extracting the components of interest.
Yeah, I think there are a bunch of companies working on this. A few companies which have recently raised a few rounds of funding are also working on using mycelia to produce. For example, there is this a Spanish startup company, Inomai, which is using fungal mycelium to basically replicate the experience of meat. So obviously, there is a lot of potential which can be plugged into the usage of mycelium to produce meat. In fact, you might have also want to use certain binders as well, right? Around these mycelia in order to mimic the structure of how the actual meat looks like in terms of its muscle fibers. Probably apply some of these extrusion techniques as well, and how that plugs in into fermentation is also very interesting. But yeah, thanks for answering that. So what role can bioinformatics play in finding novel strains of fungi?
Yeah. Yeah, I see this question, and I also see a question on how to use machine learning models to screen for fungal proteins. And I would say that this, like, using bioinformatics, machine learning, and artificial intelligence techniques are very much required in this sector, and not just for fungus, but also for other screening other microorganisms. So this is true for example, microalgae as well as bacterial species. So I believe that uh, like using this as a technique, like for high throughput screening, would one, like, help you get around like the GMO component of it, if you don't want to go around, like, go for genetic modification of strains. And also in terms of like a wide space opportunity, because right now I believe no one is doing this in India. So it would be great to have to see one of uh, these as something which plugs into the ecosystem and helps other companies which are looking for these services to actually venture into creating these products, because this can be a very costly process for companies to undergo on their own. So if there are services which could just plug into existing companies, they will be incredibly valuable.
Right. Completely agree with that. Um, let's move to the next question. So I think there are a couple of questions which revolve around machine learning as well as bioinformatics. So what are your thoughts on using machine learning models to screen fungal proteins? Is there a demand for is there a demand, or do you think there is a demand for such a service by startups or companies at this point in time? Probably you can answer this in terms of how such companies could play a role in the future and how the field is actually coming up. For example, before we had this webinar, I think you're saying that there are a bunch of these fermentation companies, fermentation everywhere in the US. So yeah.
Yeah, I think I just answered this in the context of bioinformatics. I'll just reiterate and say that it's a very valuable service. And I mean, I don't remember the name of the companies right off my head, but there are companies which are using like they have built these platforms where the only service offering is that they are able to do um, high throughput screening for these. So yes, this would be a valuable service, and like I would urge people to explore this.
Yeah, I think, and also to the participants, I think just to add to what Rama said, I think in terms of, you know, basically screening for, let's say, new strains of fungi, I think a combinatorial approach would be much better. I think there are several publications which suggest that, I mean, just based on the morphology is a weak approach, but combining that with, let's say, DNA barcoding, or let's say you find specific DNA sequences, for example, the ITS region, which is the official DNA barcoding marker for at the species levels, not only for fungi, but also for, let's say, plants as well. So obviously use some of these tools, and there are specific bioinformatic tools which uses machine learning algorithms. One of them is ITS2 BEC, for example. So probably people can use all these biotechnology methods and see which is the best approach using like 2 or 3 or I mean, the more approaches you use in combination, I think you'll be in a much more better position to find the right, let's say, species of microorganisms, not just fungi, but it could be yeast or whatever it is. Okay. Yeah.
Just one more question here, Akshay. So I think in general, Rama, if you want to talk to the strategic analysis and what all has been covered, it has been given as a resource. We'll again resend it to all the candidates today, but perhaps just having a little bit of context setting on what perhaps people can derive from it if they give it a read would be quite helpful, maybe in 5 minutes. And, you know, that will be helpful for the participants watching this recording as well.
Yeah, so I think like this slide, the presentation I gave is basically a condensed version of the strategic analysis that we did, and the strategic analysis was all about doing a technology deep dive into how like fungi can be used for alternative protein applications, and what if there are current use cases, that how they are doing this. And I think you would get more information on, for example, specifics of strain selection or like bioreactor designs, how Quan made their first product, what technology did they use, and any other public information which is basically available on any other company doing work in this space. I think some additional information on regulatory aspects, which Radhika and Satvika, who are the regulatory experts in GFI India, have had compiled, is also there in the report. And also in terms of talent pool, maybe not so relevant for the participants, but just to understand and get a sense of what kind of streams and what kind of educational backgrounds are most useful for actually creating ventures in this space, which may be more relevant in terms of if you're looking for co-founders and building your teams. So it gives an understanding of like the entire value chain, but like in much more detail. And also towards the end, we talk about how what is the maturity of these applications globally, and what is the maturity of each of the each of these application areas, particularly in India. So this can help you identify like where there are gaps, where you think you should probably focus on and put your investments into. And I think reading the report would probably answer many of these questions that we see in the Q&A section also. So definitely recommend doing that.
Perfect. Thank you so much, Rama. And again, just adding to the talent piece, I think even for founders and equally for researchers and students thinking about careers in the sector, or maybe 5 years down the line, 10 years down the line, if you want to start a company in this domain, based on the kind of challenges you're undertaking to solve them right now as part of the challenge, it's quite important to understand these opportunity areas and perhaps develop your career accordingly for the next few years, because ultimately this is a tech forward area where investors would kind of look at the founder's background and the core team's background in understanding the key specific issues that your startup is looking to solve for as a scaled up solution. So yeah, just that. Akshay, you want to continue with the questions?
Yeah, thanks, Shardul, for that. Thanks, Rama. Okay, I was just looking at some of the chat queries, but
Akshay, can I just like address like I think genetic modification again? Like I think there's some bits on regulatory, and we did not cover that in the webinar. So I think regulatory wise, again, I think I don't know, Shardul, Akshay, you have covered this in other webinars, but use of genetically modified organisms, even if you extract components out of it without using the organism itself, is still a tricky situation in India. So there is less clarity on whether there'll be approvals and how the public perception will form on genetically modified organisms in India. So I would say that it's a gray area, and probably like better to be cautious than it depends again on what application area you're looking at. So for example, for something like Rishkol Biosciences or Lazarus Bio, which is making growth factors, this is a critical thing, and the way they are looking at it is exporting these ingredients to other cultivated meat companies abroad. So from that perspective, doing genetic modification is not a problem for them because their market is not India. But if you're looking at marketing these in India and like creating end products out of it, this might be a tricky pathway to navigate. At the same time, when it comes to getting regulatory approvals for end products, so I think someone asked about like which strains are approved as edible fungi, and I think I wish Radhika was here to answer this, but we have very few of the edible mushrooms which the government actually says like in FSSAI in terms of describing which are approved. But at the same time, there are many farmers which are using a number of strains to cultivate mushrooms, and they're able to sell in the market. So there's less clarity, but we see that if you're using mushrooms as food ingredients, it should probably not be that big an issue. But I would again defer it to my colleagues to give you more clarity if you need more information.
Yeah, thank you, Rama. I've also to your question as well, I've also added some information that we have from our, again, strategic analyses report that our regulatory specialists put together. So I would again encourage you to go through that report. There's a list of strains that have been approved by the US FDA, and that's usually something that you can refer to if you're looking for any sort of pre-market approvals within the Indian market. And like Rama said, food applications are also covered by the FSSAI. So I just encourage you to read through that report, read through that regulatory section, and some of your questions may be answered.
Yeah, thanks, Nicole. Thanks, Rama. Yeah, I think we have mostly covered all these questions for today. I think there are specific questions about liability and also in terms of what role can medicinal mushrooms like shiitake play in the alternative protein industry. Shiitake is being used by multiple companies. please type in, search for these companies. Shiitake mushrooms are being used to produce alternative meat products as well. So what form of packaging? Probably we don't have the specific expertise on what kind of packaging we might want to use for specifically biomass fermentation. But I mean, if people have very specific questions, please feel free to write to us, and we'll try to find an answer for all these questions. But yeah, I think we are quite early. But if there are any questions or anyone wants to ask questions live, we can go ahead with that. Please raise your hands if someone wants to ask a question live. We'll wait for a minute.
Yeah, thank you so much for
You
the
can ask your question. Yeah.
yeah. Thank you so much for the presentation, Rama and Akshay. It was very nice hearing your thoughts. I am Arman and I'm working on biomass fermentation. And I just had a very simple question. So in school, we had always learned of hybrid plants. But do hybrid species of mushrooms exist in any form? I know you've talked about inducing mutations to increase certain properties, but do hybrid species of mushrooms exist? Because they reproduce through spores. So I just wanted to know if there's something known as hybrid mushroom.
I'm hearing this for the first time, so again, like not sure. What do you mean by hybrid mushroom? Like, what is it that you
Right.
have in mind?
Sure. So, is there any approach where we can possibly, let's say, take the properties of shiitake mushroom and then take the properties of maitake mushroom to produce a new variety of mushroom?
Yeah. At least like first of all, like I'm not an expert on like genetics aspects of it, so I have not come across this, but I would probably like encourage you to like look at other experts and maybe like Akshay, if we have someone you can recommend.
So I just
Sure,
did a
Arman. Yeah.
Google search. I found a few things. Maybe Akshay is adding to it already, but I found a few papers where hybrid strains are basically used to create desirable characteristics in the end mushroom product. So I again, not an expert on this, but perhaps those papers might help you, Arman, further. But Akshay, go on.
Yeah, I mean, just seconding what Rama and Shadil said, but obviously, I think when you talk about hybrid mushrooms, I think the basic question here to ask is whether you're considering producing hybrid mushrooms, for example, in a laboratory, or you're looking for naturally produced hybrid mushrooms which occur naturally, for example, finding new species which has occurred in nature, or which have not been discovered before. In either case, I think probably also tying this back into how regulatory aspects look into this. For example, if you're trying to get something out through human intervention, I think that's a different story versus if you find something which is naturally occurring. So yeah, I mean, obviously, like Shadil pointed, there are several publications which point towards or which describe some research done on hybrid mushrooms, for example, oyster mushrooms. So probably I think you guys can find it. And in case you guys would like to access these publications, do let us know.
Right. And thank you so much. It was very helpful.
Thanks, Arman.
Okay,
Henry
you
has.
wanna ask a question?
Henry, we can't hear you.
Okay, I think I am in.
Hi,
Yeah,
Henry.
hi Rama and Akash. Thanks for this kind of webinar and very good session. Actually, I want to ask about the mycelia biomass can be hybrid with the whole mushroom for the improving of textures as well as the umami. Any idea?
I think like I have not come across this, but I think it would be a great great thing to explore. Like honestly, did not see any applications where they combined biomass fermentation with whole biomass, but I can see that happening because like, you can potentially like improve the quality of either biomass fermentation as well as like whole biomass fermentation through those routes. I would probably like be a bit aware of like the engineering challenges, like especially with scaling up this kind of a process. But yeah, I think that looks like an interesting idea. Henry, are you working on it?
Okay, thank you so much. Yeah, we are working on that, and about it will come about 60% of the mycelium, and other 40% will become from whole mushroom.
Mm-hmm.
I think Akash will understand. Akash, Akshay, sorry, Akshay.
Actually, yeah, no worries.
Yeah,
Thanks for your question, Henry. So I think I tried to answer it. Probably, I don't know whether you had a look at my response to your question. Uh, Companies do consider using mycelium to produce those muscle fiber-like structures, and probably mushroom can form that soft, meat-like texture as well as. I mean, it's a great experiment to explore. But if you do come up with something like this, an innovative product, do let us know. But obviously, I think what in very simple terms, probably mycelium could be mimicked as collagen, and mushroom as the soft texture for meat.
yeah. I probably it will come after it in formulation part, in engineering part, as Rama said. It will come into
Yeah,
formulation part. Both
yeah,
of
yeah,
them combination will come for textures as well aroma, because the both biomass itself is not means from our experience, biomass itself is not completely for the midlife structure for ourselves, and combined with the whole mushroom, it contain with fibers. It I mean double the fibers itself.
yeah,
That's why I just asked
yeah,
very kind of things that very specific and very unique things. Thanks for answering, Ramya and Akash.
yeah.
Yeah,
Thank you.
thanks, Henry, and probably 1 or 2 years down the line, we can have you as an expert giving a webinar on biomass fermentation, probably. But thanks for your question, Henry. Uh,
Sure, sure.
uh, and we are getting very different types of questions now. Do you see a combination of fungi and bacteria being used in precision fermentation?
I mean, you can
It's
use it.
fungi.
Like, there's no limit to what you can do with science. Um, I mean, I think the question to ask is like, why do you want to do it at first place? Like, what is the additional benefit that you're getting out of it? Uh, and also at the same time, I mean, if there are symbiotic benefits with using fungus and bacteria, like maybe that's a route to go. You would again probably need to look at the same things, which is strain optimization, bioreactor design, uh, making sure the bioprocess is conducive for the growth of microorganism, and I think like you will have to get back to the question of like, what is it that you want to achieve out of this? Like, is it just because like it's a science experiment, or is it because you're seeing that uh, for example, you're the fungi is consuming the bacteria, or like other way around, and that is improving the quality of protein that you're getting. So again, like interesting areas to explore, but at the same time, keeping an eye on the practical aspects of whether something could be done or not. And I obviously like I just want to say that I'm not an expert uh, in biology at all. I have a chemical engineering background, and I really urge everyone to like do search on like Google Scholar. It has been very helpful for me to understand things that I have no expertise on. So definitely encourage, if there are answers I'm not able to clearly give, please understand that it's less because I don't want to give, but more because I probably don't have the entire knowledge and I don't want to give the wrong idea.
Just a quick
Yeah,
thought here
thanks, Ram.
for
Yeah.
just a quick thought here, Akshay, for track 2 participants, it's a very, very relevant point, right? Doing secondary research on the right platforms like Google Scholar, ResearchGate. It's just a matter of putting in the simple keywords that you want to understand more about, and papers across the years pop up, right? And you're able to see how the technology has progressed. And, you know, a lot of scientists and researchers and students who are academically inclined might already know this, but also saying this for founders who don't have a specific background, perhaps in fermentation or cultivated or plant-based kind of tech-forward areas. It's very, very easy to acquire those, set of data points or at least information sets from these platforms. And then perhaps you are able to think about your own idea and talk with your teammates who might be from a scientific background at a much better level. And perhaps the overall quality of proposal just drastically improves if you follow that path. So yeah, just a little simple something.
Yeah, thanks, Shadul. And also, I think what Rama pointed out in terms of, for example, for the last question, what are you actually thinking about? Is it just a science experiment, or you're actually building a proposal, for example, in track 1 and track 2, and ultimately what you want to get at the end of this proposal is something which is solving a white space area in this space, or bringing new innovation, for example, probably in taste or texture, a new product on the market, or a new technology which brings down the cost of producing these products. So I think that your mindset should be in such a way that it's not just about combining different platforms and seeing if we can produce something like a very exciting scientific experiment, or at the end of the day, probably you should also think about that, okay, my proposal is solving something big in this sector. And once you get there, I think that's how a really good proposal will ultimately look like.
Yeah, I just want to reiterate what Akshay said. In any problem solving process, and it doesn't matter whether you're doing a business problem solving or a technical scientific problem solving, always start with what is the gap that exists, what is the problem that you want to address, instead of saying, oh, I want to, like, I have this great idea and let's try to figure out how it can solve something. That may or may not work, and probably like you would end up spending a lot of time pursuing something which may amount to nothing. And I think this is something which is very key to like design thinking processes, which I highly recommend if anyone like wants to look at like how to be more efficient and effective in any problem solving process. Is start with the need. Start with what is the need out there, be it a business need or scientific need, and then go from there to finding out potential solutions. Then keep your mind open to all the different kinds of ideas that can solve a particular problem, and then like choose what you want to work on based on like your particular boundaries and constraints.
Thank you, Rama. And thank you, Carl, for sharing that. Carl, I think earlier you had your hand raised. Would you like to ask a question? It was a few minutes ago, though. If you do, you can just raise your hand. Or if anybody else has any question, we still have 5 more minutes. You can quickly raise your hand and we can promote you as a speaker.
Thanks. Shadul, probably you can close it out.
Yeah, okay. Thank you so much. So I think we don't have any more questions, but thank you so much, Akshay and Rama, for taking the time out today and sharing your thoughts on the fermentation and, especially the fungi side of things when it comes to our technology areas and focus areas. I think this will be quite helpful for participants thinking about these applications and, the material that we'll kind of send across with this. Make sure that you go through it. And do join us for the webinar tomorrow as well, which will be focusing on plant-based eggs. Tell your teammates about it, but largely it's also going to talk a lot about ingredients, fats, new product development processes, R&D challenges when creating some of these products, which are applicable to plant-based meat, dairy, and seafood as well. So tune in tomorrow 6 p.m. for webinar number 5. And once again, thank you so much, Rama, for this wonderful webinar, and Akshay for moderating it. Thank you, Nicole, as well, for the help with the questions and moderation support. Have a great day, everyone. Bye.
Thank you. Thank you, everyone. Bye.